This transcriptomic analysis of human prostate cancer tissues identifies individual human endogenous retrovirus expression loci to evaluate a custom high-density microarray as a screening tool for biomarker discovery. After the surgeon has removed the prostate organ from the patient, the pathologist prepares tumoral and adjacent normal tissues separately. In the lab, extract, purify, and qualify mRNA from the normal and tumoral tissues. Amplify mRNAs using whole transcriptome ovation kit and then cleave and label the resulting amplified products. Then, sequentially process the HERV V2 microarray by filling, hybridizing, washing, and scanning. Ultimately, using biocomputing methods, probe sets exhibiting significant signal and differential expression can be tracked, leading to the identification of transcriptionally active individual loci.
Many years ago, we explored the behavior of the HERV family in various contexts, including multiple sclerosis samples, placenta, testes. I first had the idea for this method when we began to understand that overlapping or non-overlapping subgroups of HERV elements within a family were expressed depending on the context.
Using chip technology allows for a coordinated exploration of several HERV families and the simultaneous analysis of different regions for each locus. For example, U3 and U5 domain for LTR, which may support a direct role in the pathology.
This method can help answer key questions in the cancer field, such as